Hello everyone, this is Harsh Bassi, your biology educator, and today in this session we will be starting with the another lesson for of genetics, in which we are going to cover polygenic inheritance. But wait, before that I want everyone to comment down and mark your attendance. Okay, now, and in case of any doubts, you need to comment those doubts in the comment section. I will be taking up those doubts and I will be solving those doubts in the comment section only. So let's quickly start this session. So polygenic inheritance. Polygenic inheritance is what? Polygenic inheritance is also known as quantitative inheritance. Now, sir, this quantitative term is kind of related to quantity, right? So, yes, my dear child, this quantitative is something related to quantity only. Like here, what happens? The quantity of dominant allele matters. Okay, so we'll figure it out, like what actually happening in this polygenic inheritance. First of all, let us know that who discovered this. So the scientist name Nelson discovered this, and this was discovered in the kernel color of wheat. He discovered it in kernel color of wheat. Okay, clear to everyone? Great. Now, what happens here, sir, that we should know? Yes, obviously. So see, in multiple allelism, what was happening? There was like more than two alleles were controlling one character. Yes or no? Yes. But here, what's happening? One character is controlled by more than two genes. Sorry, alleles. Okay, more than two genes. If I am using the word gene, so I should write genes pairs, and if I am using the word allele, so I'll just write here allele. Okay, clear? Any doubts anywhere? Polygenic inheritance, the definition, I think so it's very much clear to you all. Okay, I'll not write it here. Okay, I think so it will be not visible to you, so I'll write in this part only, like this part only. Okay, great. Chalo. So now let's have an example. This polygenic inheritance or quantitative inheritance has a very common example, which is what? Which is human skin color. Okay, and this example only you have to learn. Example is human skin color. Okay, so see, human skin color is basically what? It is controlled by more than three alleles. Okay, or I should say more than three pair of genes. And if I'm using the word allele, I should not say three, rather I should have said six. Okay, so controlled by more than or two alleles, which is controlled by three alleles. Why I'm not directly writing, God knows. So controlled by three genes, or I can say six alleles, right? Yes or no? Yes, sir. This is very much perfect, no doubts. Okay, chalo. Now let's move forward and let's us know that what are the genes which are controlling the human skin color and how it is controlling, right? So here, if I talk about capital A, one gene, capital B, the other gene, and capital C, the other gene. If I talk about their alleles, so it will be what? Small a also, small b also, and small c also. So these are the genes, these are the three types of genes which is controlling the human skin color. And these are what? These are the alleles of a single gene. So in total, three genes are there and six alleles are there. 1, 2, 3, 4, 5, and 6. Clear? Okay, sir. This much is clear. Now let us understand this human skin color in much detail. Let us perform a cross. Let's understand this with the help of a cross. See, so let's take a full dominant one, AA, BB, and CC. So this is what? This is a full dominant one. Yes or no? Yes, sir. This is a full dominant one. Now, do you understand if I say that this is a full dominant one? So as of now, you remember that if all the alleles are dominant, the person will be black, darkest black. Okay, person will be darkest black, or I should say full black. The person will be full black. Yes or no? Okay. Next, what, sir? Next, let's take about the another one, which is having pure recessive gene, and this pure recessive gene will have the full white color or full fair. Okay, full white, or I should say full fair. Clear to everyone? Any doubts? Any doubts till now? No? Great. Now let's move forward, and I have taught you how to make the gametes and all. Yes, so make the gametes, and here you are going to get what? Here you are going to get gametes as capital A, capital B and capital C, and here you are going to get gametes as small a, small b, small c. Yes or no? Use the fork line method now. And when these are gonna be crossed, so we are going to get this color, this one, this genotype. Yes or no? Yes. Now see, here what's happening, three of them is dominant and three of them is recessive. So in this case, the color will be what? The color will be intermediate, or it is also known as mulatto. Okay, clear? Great, sir. Now again, what we are doing is that we are performing the F2 cross. We are performing the F2 cross. In humans, we can't do the sibling mating, so we might have find from somewhere the the female or the male of the same genotype. And when we do the crossing, we get what? We get some ratios, and the ratios are what? The ratios are 1:6:2, 15:2, 20:2, 15:2, 6:1. Okay, this is the ratio which we are getting when we are performing the F2 cross. So this was what? This was the F1 cross. The blue one was F1 cross, and then we did, when we crossed like same genotype ka cross humne karaya, and then we got some ratio, and this will be called as what? F2 phenotypic ratio. Okay, this is what? F2 phenotypic ratio. Sir, do we have to learn this? Yes, bacha, definitely. So see, we got the ratio 1:6:2, 15:20 15:6:1. See, this much you learned, just this much you learned. Baad mein to it's same. It is same. Okay, clear? Chalo, great, sir. Now the ratio one which we are having will be what? Here the genotype will be, or rather than genotype, I should say the phenotype will be full dark phenotype. This will be what? In this, what is happening? In this, the color will be very dark brown, and here what's happening in this case, if I talk about this case, so in this case, all the alleles are what? All the alleles are dominant. In this case, 5 of them are dominant. Okay. In this, what's happening? If I talk about the genotype, so the genotype for, the phenotype for this is what? Dark brown. And the number of dominant alleles present here is what? It is just 4. Next, I talk about this thing which is 20. So 20 is having what? In this, 3 of the alleles are dominant, and the color is intermediate. The skin color is what? Here it is intermediate. Okay. Next, what? Only 2 of them is dominant, and in this case, slightly mulatto. Okay, then we have this. So in this case, only 1 is dominant, so less white, and this is no dominant, 0. All the alleles are recessive, so it will be full fair or full white. So can you see, as the number of can you see here that as the number of dominant allele is decreasing, the color is getting light and light. Clear? So this is what this is the quantitative inheritance or polygenic inheritance. If with respect to polygenic inheritance, if I define this, so you may understand that a gene controlling, a single character is controlled by more than one gene. If you want to understand this example with the quantitative inheritance, so if the number of alleles, basically number of alleles is affecting the phenotype, number of dominant alleles is affecting the phenotype. Okay, clear to everyone? Any doubts anywhere in the right now? Let me know in the comment section. No doubts? No doubts? Great. Okay, good. Now let's have a question. There is a very famous question which is usually asked on this topic. So the question is, let's suppose if the plant height, for example, if the plant height is controlled by two genes, okay, by two genes, clear? And maximum plant height which the plant can have is, let's suppose, 30 centimeter, and maximum plant height can be 30 centimeter, while minimum, how much? Minimum is 20, while minimum plant height, can be what? Can be 20 centimeter. Okay, then what must be the, then what should I say? Then what must be the plant height of the following genotypes, Okay, so the genotypes which we have in the question is capital A, small a, capital B, capital B. Let's have capital A, small a, capital B, small b, and let's have one more which is capital A, small b, small b. So how we can solve these types of questions? See, basically it's saying that this is the minimum height, this is the maximum height. Now calculate the phenotype of other genotypes, whatever is given in the question. Okay, so this is a question of polygenic inheritance, and how we can do is that, see, first of all you need to understand that how much one dominant gene is contributing, right? If somehow we are able to understand it, we can figure out something, yes or no? So see, how we can say, first we are going to calculate contribution of one dominant allele, okay, contribution of one dominant allele. So the contribution of one dominant allele will be what? How we can calculate, sir? So we can calculate it by maximum phenotype minus minimum phenotype, okay, maximum phenotype minus minimum phenotype upon, divided by maximum number of dominant alleles, maximum number of dominant alleles, like whatever the maximum number of dominant allele is possible, you have to write. So sir, here what's there in the question? It's saying that 30 centimeter is the maximum height and 20 centimeter is the minimum height, yes or no? So I will go with 30 minus 20 upon maximum allele possible, maximum dominant allele here possible will be what? See, two genes are controlling the character, they said, okay, they said that two genes are controlling. So maximum, how many dominant alleles we can have? 4, okay, divided by 4. अगर तुम्हें अच्छी सी मैथ्स आती हो, if you are good in maths, so 30 minus 20 is 40. No, 30 minus 20 is 10. So 10 divided by 4 will be 2.5. That means each allele is contributing how much? Which is 2.5. Okay, sir, we got it. Now, how to calculate the phenotype, sir? See, now phenotype, how we will calculate. So what you have to do is that minimum phenotype, you have to write minimum phenotype. See, you have to learn the formula for this, right? Plus contribution of one gene, contribution of one gene multiplied by number of dominant alleles in a given genotype. Number of, हो मैंने अनअकैडमी के लोगों पे ऑफ लिख दिया, कोई बात नहीं. Number of alleles in dominant what should I say, dominant genotype. Okay, now see, things which we have written, we have been asked to calculate this. So let's write here capital A, small a, capital B. Was it small b or capital B? Capital B only. So in this case, how many minimum phenotype is what? Minimum phenotype we have said, we have been said about is 20. So 20 multiplied by 2.5 contribution of one gene. Sir, ये कहाँ से आया? ये यहाँ से आया बच्चों, अभी निकाला हमने. 2.5 multiplied by number of alleles in dominant genotype. So number of alleles in dominant genotype will be what? It will be 1, 2, 3. So multiplied by 3. And if my maths is okay, so 2.5 multiplied by 3, 5 which is 7.5. Okay, and 7.5 multiplied by 20. So I remember that 10 or point wala hat jayega, and then we will get what? Oh, sorry, bachcho, sorry. Here it will be plus. It will be plus. Okay, it will be plus, not multiplication. Okay, it will be plus. So here the phenotype will be, the height will be 27.5 centimeter. Okay, similarly you have to calculate this one and this one, and let me know in the comment section. Okay, clear? Chalo, great. In this way I will be able to figure it out that how many of you have watched the complete video. So those who are commenting, I will be like, okay, you have watched the video, and if not, then I'll punish you. Okay, chalo. Now let's move on. And let's talk about the another thing, which is cytoplasmic inheritance. So in polygenic inheritance we have to study this much only. Now let's come to the another one, which is which is the cytoplasmic inheritance, the next topic. Okay, sir. Let's talk about the cytoplasmic inheritance. So cytoplasmic inheritance is what, sir? It is just because of the gene which is present in the cytoplasm, not in the nucleus. Okay, great. Now we all know that we have egg cell, right? And we have sperm. Yes or no? In which one the cytoplasm is present in more amount? In which one there is the chances of presence of gene in the cytoplasm? See, sperm is so small. Sperm doesn't want to keep anything extra. Sperm wants to keep itself very light, so that it can flow very rapidly and go inside the egg cell, so to fuse. Yes or no? Yes, sir. So in sperm we can't expect the other cell organelles, many cell organelles, and genes also. So in egg only there is the presence of cytoplasmic gene. Okay, clear, sir? So here gene is present where? Gene is present in cytoplasm, and if that gene is affecting the phenotype, then it is known as cytoplasmic inheritance. Gene is present in the cytoplasm, affecting the phenotype, So in this case we will be saying that cytoplasmic inheritance is happening. Said, where the gene will be present in cytoplasm? Maybe the mitochondrial gene, maybe the plastid genes. Okay, clear? Chalo. So now what is going to happen? See, whatever genes, wherever it is present, but if it is present in the cytoplasm outside the nucleus, those genes are known as what? Those genes are known as plasma genes. So you can write here all genes all genes present in the cytoplasm, all genes present in the cytoplasm is known as plasma gene. Okay, is known as plasma gene. Clear to everyone? Great. And what are the genes present in the nucleus called? So genes present in the nucleus is called as karyogene. Okay, karyogene. Any doubts anywhere? Let me know in the comment section. No doubts? Great, great. If you have any doubts, please comment in the comment section. Okay, great. Now, cytogenes are what? If I talk about the plasma gene, so I can also give it one more name as cytogenes. Yes. Example of cytogenes, mitochondrial genes or genes present in the plastids, etc. Yes or no? Great. Now, see, egg is present where? Egg is the part of the mother, maternal thing, right? So can I say that cytoplasmic inheritance can also be called as maternal inheritance? Why? Because it will be inherited from the mother to the babies, right? There will be no role in the sperm here. Clear to everyone? Okay. So can I write here cytogenes or the plasma genes? Cytogenes or plasma genes, what they show? They shows maternal inheritance. Okay, maternal inheritance. Clear? Okay, sir. And see, whatever the phenotype will be present here in maternal, it will be reflected in their babies. So whenever we are analyzing the pedigree charts or something like that, so if you see that suppose mother is in green color, and if mother is in green color, all the progenies are green, so you can have a think of this cytoplasmic inheritance also. If mother is tall and all the babies are tall, irrespective of what the father is, then also you can think about the cytoplasmic inheritance. Okay, okay, sir. Now let's talk and let's understand this better with the help of some examples. So let's have the example of Mirabilis jalapa plant. Okay, we have also discussed one example. Where we have discussed, let me know in the comment section. Okay, so we have one example which is Mirabilis jalapa katappa. No, jalappa, sir. Where are you, sir? Why katappa? There was a very famous dialogue, right? Bahubali ne katappa ko kyun mara? Something like that, right? Katappa ne Bahubali ko kyun mara? No, no, no. Bahubali ne katappa ko kyun mara? Okay, chalo. Now, Mirabilis jalapa. So in Mirabilis jalapa, we have three types of leaves. Okay, we have three types of leaves. One is what? One is green leaves. Second one is what? Pale leaf. Pale color is like little yellowish color. And the second is what? Third is the variegated leaf. Okay, like दोनों का mix and match. थोड़ा सा तुम ले लो, थोड़ा सा मैं ले लेता हूँ. तो variegated leaf. Clear? So here what's gonna happen, guys? See, in green leaves, see, obviously, in green leaves there will be what? There will be like these pigments. Okay, the green pigments will be present. In pale leaves, the pigments will be what? Of yellow color. Okay, so let me make some yellow color. Oh, why I'm using this highlighter? Let me use some yellow color here. In variegated leaves, what is going to happen? The pigment will be kind of distributed like this. Somewhere green, 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 somewhere yellow, 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 or pale color, pale color, pale color, pale color. Okay, clear? Understood? Any doubts? Now what? We are doing the cross. We are doing the cross. Which cross, sir? Between the female, which is green in color, and male, which is pale in color. Okay, so female flower, which is green in color, and a male flower, a male parent, which is pale in color. But whatever happens, the child will be green. Why? Because female is green. In a similar manner, let's have the another cross. Do I have a space here? Yes, sir. This much space is there, sir. Why you are not able to see, huh? Why does this behavior, sir? See here, cross 2, cross 2. So cross 2 will be what? Here we have female, which is now pale, and male, which is green. Male, which is green. Okay. So when we are going to cross them, what we're going to get? We are going to get pale colored progenies. Okay. So can you see whatever the female genotype, phenotype is, the progeny will have that phenotype only. There will be no change in them. Okay. Clear to everyone? Great, sir. Now, this was the example of the this was the example of what? This was the example of cytoplasmic inheritance. Clear? Other cases of cytoplasmic inheritance, what cases we have to remember? Other cases of cytoplasmic inheritance. Cytoplasmic inheritance. Okay, 1 second. Before that, I'll also explain you one thing. There can be one more situation. Na? Ek aur situation when we are studying, let's study only. So we have what will happen when female is variegated. When female is variegated and the male is, let's suppose, pale or any other color. Okay, so see, to learn this, how what things can happen, see here. If suppose I ask, this inner circle represents the color of the female, okay? I need 3 colors here. Arey sir, what you are doing? 1 second, guys. Thoda. Tung, tung, tung. Mercedes, Benz Mercedes. No, I'm not promoting the brand. Don't worry. So see here. We have green color, we have pale color, and we have variegated color. So what can be the chances here? This inner circle represents what? This represents the female characters, okay? This represents the female. Now let's have the outer characters. Outer circle, okay? Let's make one more circle. And in this circle, let us have the male characters, okay? So male can be what? Male can be what? It can be pale in color, it can be green in color, or it can be variegated. Yes or no? It can be variegated. Yes. But what will be the color of progeny? What will be the color of progeny? So whatever happens, the progeny will be what? The progeny will be green in color. Why? Because the female is green. Next, let's go with the pale color. So in pale color also, we can have what? We can have pale male flower, we can have green male flower, and we can have variegated male flower. Yes or no? Yes. So, but the progeny will be what, sir? The progeny will be, in this case, in this particular case, where the female is pale, so the progeny of will be also pale. Yes or no? But if I talk about this thing, where the female is variegated, so male can be what? Male can be green, male can be pale, male can be violet, but whatever happens, the progeny formed by the variegated female and these when crossed with green, pale or variegated will be what? See, it can be, here the chances can be what? The chances can be of green variegated and pale, okay? Then the chances will be what? It can be green, it can be pale, it can be variegated. In a similar manner, here also it can be green, it can be pale, or it can be variegated. Again, it can be green, it can be pale, it can be variegated. Okay. So if the female is variegated, so the chances will then the cross can have green color also, can have a pale color also, can have variegated also. But if the female is pale, it will only have pale. If the female is green, it will only have green. Yes or no? Clear to everyone? Okay, so in this way you can remember what is the condition. If the female is pale and the male is pale, then what color is going to have? If the female is green and the male is pale, the progeny will have green color, yes or no? Right? If the female is variegated, then possibilities are there that the progeny can be green, it can be pale, or it can be violet. Okay, so during the variegated condition we have the other possibilities also, but when the female is green or just pale, the other possibilities are zero. If the female is green, it will be, the progeny will be green. If the female is pale, the progeny will be pale. If the female is variegated, then the progeny can be green, pale, or variegated. Okay, clear? Okay, sir. Any doubts? If there is any doubts, let me know in the comment section. Okay, if you are making the notes, make this thing in a beautiful circle, okay, and share it with me. Chalo. Now, other cases of cytoplasmic inheritance, we don't have to remember, or we don't have to study about the crosses, but just for the sake, let's do what? Let us learn the examples. So male sterility, one example is male sterility in maize. Okay, male sterility in maize. Second will be what? Second is let's have Pochineurospora. Okay, Pochineurospora. If the neurospora is pocky, like pokes are there, Pochineurospora condition is also the cytoplasmic inheritance. Okay, and let's have IOJAP color, IOJAP inheritance in maize. What is this? Green and white stripes. Okay, green and white stripes in maize. Okay, learn these three examples which can be asked in exam. Okay, but the chances are very less. But see, again, lots and lots of syllabus has been reduced, so genetics might become tough. Might be that the NMC is, or the NTA wants you to have tough questions in genetics, right? So green and white strips, this pattern is known as IOJAP pattern. IOJAP pattern. Clear? Okay. Any doubts anywhere? Let me know in the comment section. So this was all about the cytoplasmic inheritance and what, and this polygenic inheritance. Okay, so tomorrow again we are going to complete linkage, which is chromosomal theory of inheritance, and then sex determination and sex linked inheritance. After this chapter will be complete, and then when the chapter is going to be complete, you have a surprise on 24th, right? I'll give you the hint. The hint is, you were demanding about some things and that demand is completed, right? Okay, chalo. So bye bye everyone, see you in the next class. And yeah, you can download the notes from that Excel sheet. Okay, I will share that Excel sheet link in the comment section. Okay, chalo. So bye bye everyone, see you.